Genome engineering is really important because you've got all of the pathway genes that the cell already has on this genome that is not typically as accessible to us as something like a plasmid. So we started off in the course talking about the ease and convenience of plasmids and how part of the reason we dove into some molecular cloning is this idea that, okay, we want to do that process typically outside of a cell, because especially if we are trying to manipulate small things and introduce new components with high fidelity, we want to be able to piece those together on something like a plasmid. And then we also want the ability to do selection, to have something like an antibiotic resistance marker to know what's gotten the change. And those questions come back somewhat on steroids when it comes to genome engineering, because a lot of the changes that we talked about, even in the examples of the dynamic metabolic control, usually we're talking about turning off native pathways. And that's not necessarily so straightforward because if you've got just, say, your one valve, okay, that's better than having to turn off 3 or 4 things. But in at least one of the examples we talked about, you still have to knock out or control 3 or 4 things. And if you have a whole pathway that you want to manipulate, maybe it's expression level, what have you, say your metabolic flux approach, your flux balance analysis says, hey, you need to upregulate flux through this whole pathway, and it's going to require you to essentially give these 4 genes this upregulation of some kind, so maybe a stronger RBS. Well, you've got to get into the genome to do that. Or, I mean, if it's just upregulation, actually, I take that back. There you could just introduce another copy on a plasmid. But if you got to downregulate, that's especially a case where you need to you got to mess with what's already there. So that's what we'll talk about now for the remainder of this lecture. First, the tools, and then applications. And any discussion of genome engineering, I think, should also start at a high level and think about concepts that aren't necessarily specific to metabolic engineering, but are all the rage in modern biotechnology, which is this general interest in genome editing technologies for human health in particular. Has broad societal consequences, ethical, moral. We'll talk about that a little bit. And as we talk about those, a major takeaway here is that actually a lot of those tools aren't going to be so commonly used or useful for metabolic engineering, mostly because of the organisms that we choose to work with as model microbes, or even some non-model microbes still aren't necessarily the most compatible with the kinds of genome editing that we might want to do in humans. And that's exactly this third point, which is that the ease and method of genome engineering is highly organism dependent. We'll give some examples of that. And there are common themes. You've got to, anytime you're trying to make a change in the genome, you've got to have something that directs you there. You don't want to just be making cuts or exchanging things across the genome all over randomly. I mean, there are some viruses, for example, that or mobile elements like transposons, and maybe even some, you could argue, some engineering applications where if you don't actually know what you want to knock out or what may give you a certain phenotype, maybe you do actually want to introduce random mutations across the genome, have something like a transposon, a jumping gene of sorts, creating a library where you've just had the gene effectively change your genome randomly, and then you have some kind of screen or selection to see what won, and then you go back and try to figure out what happened. So there is some of that, but again, thinking about it more from the engineering standpoint, we usually have a particular pathway or gene that we want to affect, and so there's the directing it, whatever your change is, and then I just talked about selection. You've got to have also some way of figuring out what change you made. More importantly, which of your cells had that change. Because a very common theme related to all of these is that these methods do not, in almost all cases, they are not going to give you 100% efficiency. of changing a genome. So, with a plasmid, you can see, does it have this antibiotic resistance or not? As you're messing around with the genome, you might have a harder time, especially if you're using a method where only 1% of your cells are going to get the edit that you want. So, what does genome engineering offer society? I hinted at the therapeutic use in humans. There are a number of diseases where we have some kind of a flawed copy of a gene. And I don't think I have to go into what some of those can be. But I will mention some where we have a flawed copy, some, not all of us obviously, but those who are affected, we may have a flawed copy of a gene where it's off by only a single base pair. And diseases that come to mind that fit that description include sickle cell anemia and cystic fibrosis. These are things that we've known for a long time. Okay, this is the base that's wrong. And maybe we know even further, this is the protein then that it makes, and why that protein doesn't perform the action that it should. And maybe we have some treatments that we can use to mitigate, and oftentimes because of our sort of diploid nature, usually a patient might have one working copy of the gene. So something like sickle cell anemia, for example, you do see a difference between people who don't have a working copy at all versus people who have one working copy. And so you can tailor your treatments accordingly. And heck, we know also, at least in cancer, there are some genes, the BRCA2 gene is a really popular one that might predispose some people towards breast cancer. And so there has been in the last 10 years an increased idea of testing to see if you have that. And right now, if you have that, and you're a woman, you might be considering a double mastectomy, which, it could, a preventative approach like that could be really important, but obviously has some pretty significant consequences, right? So what if you could just take some of these technologies, deliver them to the right cells, and make that edit either within a living patient, or you take some of their cells out, reprogram them, put them back in, and the body works with them. There's a lot of interest in doing that, but once you've got an adult and you've got all this complex biology, sometimes it can actually be harder, particularly if you need to affect change within a patient throughout an entire type of tissue or something like that. And so then there's been this ethical question of doing it in the germline. And I mean, the ethical questions also around editing adults are significant too. They could be medical, like I just mentioned, or you could imagine that people start to get more interested in other traits. And so this chart here kind of highlights this idea of, do we want to be curing things or preventing, and how that could work differently in somatic cells versus germline. And so how you can have for editing, maybe the simplest example, you can target cells in particular, you can target genes in particular types of cells, and maybe you can use a virus that only infects those cells, but a safe version of a virus, engineered to not cause any other kinds of problems, to deliver something to make some kind of an edit. Or you can do that in the germline by modifying zygotes like sperm and egg. But then that change, so that change will affect all the cells in that future person, but it will also be perpetuated without their consent for generations. And so it's pretty consequential in that sense. And I think that's what's mentioned here. The heritability of germline changes is pretty significant. But yeah, we've actually been doing somatic cell therapies of various kinds for a while. So this isn't completely new in that sense. And so it's interesting as the general public, and maybe any of us, think about how do you take a tool, a platform like genome engineering or editing, and decide when it's appropriate to use it. And a big part of the perception here is governed by what the change would be used for. So I mentioned if it's a medical condition, a disease in particular, then people seem much more okay with it. But so this also depends on what kind of disease. Are we talking about treating or eliminating something really severe, possibly fatal, that would impact a person for the rest of their life? Those seem like more of a no-brainer. Okay, if we have the ability to make this kind of change, and if there are relatively few risks, then is it almost more ethically problematic to not kind of make that change? But at the same time, okay, if what we're doing is just reducing risks, should we be doing it? That seems to make a difference. And then, of course, if we're doing this to perform genetic enhancements, creating, trying to promote intelligence, that's something that starts to seem much more controversial and make